These are the lecture notes for chapter 45, which is population and community ecology. Little warning, you set this up first. Here we go. Little warning, this is probably the longest chapter that we have had. A lot of information in this chapter. So we're going to start out with population demography. So the population is just defined as all the members of the same species that live within a specific area. I'm going to add that in here. So all the members of the same species that live in a specific area. So we can talk about the population of largemouth bass in a lake, but we can't say the population of fish in a lake. So population is defined as referring to members only of the same species that live within a specific area. And then demography uses mathematical tools, so it's more graphing and statistics, to investigate how populations respond to changes in their biotic and abiotic environment. And biotic just means changes in living things, such as other species dying out or other species maybe invading the area. That would be biotic. And then abiotic conditions would be weather and availability of nutrients and minerals and water, that kind of thing. So, so whenever we talk about population demography, some of the things that are measured are population size, and you'll have to remember this, that capital N means the total number of individuals, the total population size. Population density is different. It is the size, in other words, the number of individuals, but it includes it's the number of individuals within a specific habitat or within a specific area or volume. So population density is using population size, but also the size of the habitat. So the population size may be large and the habitat is large, so population density is small. But if the population size is large and the habitat is small, then population density is higher. So population density is the number of individuals per area. And we have a graph here. So this is an example of a population-based study using population demographics. You can see all these different species. I don't even know all of these, but these are obviously from Australia. But the quoll, the bandicoot, wombat, the rat kangaroo, the potoroo, possum species. Oh, no, because possums are not. So I guess, I don't know. I don't know why they would have picked these specific ones, but I know a kangaroo and a possum are marsupials. A wallaby is also. Anyway, these are different species of organisms. The bear cuscus, and the glider species. Okay, so if you look at this graph, this shows an inverse relationship between population density and body size. So body size is the down at the bottom, the x-axis, and then the y-axis is the population density. So if you look, the higher the population density, the smaller the body size. And then the lower the population density, the larger the body size. So it's just saying larger animals have a smaller population density, which we expect. Um, this is a picture of a man that's using a quadrat to measure population size and density. Um, you obviously can't just measure, you can't just count all the individuals in an area. So you use different methods to estimate the number of individuals in an area. These are pictures of animals that have been researched using the mark and recapture method. So A is the bighorn sheep. You can see this one has been marked. B is the California condor, and C is a salmon. You can see the marking. Okay, so you mark them, and then you do a population sample um, and mark all of them, and then you do another population sample and see how many of the marked ones are in it. So mark and recapture, this is how it works. Individuals are captured, counted and marked, and then they're released. And then later, at a different time, maybe a month later, a year later, another group is captured, and you measure the ratio of marked to unmarked individuals in the second group. And that helps to estimate the total population size. So here is how mark and recapture is used mathematically. This is the calculation to calculate N, which is the population size, or estimate it. So if in the beginning we capture 80 deer and mark them, tag them, and release them, and then later we capture 100 deer and only 20 of them are marked, then this is how we estimate population size. We take the number marked in the first catch, which was 80, times the total number of the second catch, which was 100. So that's going to be 8,000. And then divide that by the number marked in the second catch, which is 20. And so we estimate the population size to be 400. Species can be distributed. They can show different patterns of distribution or dispersion. So dispersion and distribution are the same thing. And these are some examples. If you look up here, it's a little easier. So species may be distributed uniformly. So if they're uniformly distributed, you have in the habitat that you're in the region that you're looking at, you have about the same number of species. They have about the same amount of space between individuals. If it's random, then they're just randomly distributed. You have some over here and some over here and some one over here. And then clumped, they're all together in one big group. So animals that live in herds, for example. They give examples down here at the bottom. Penguins, which are territorial birds, tend to have uniform distribution because they are very they want to have their space, the same amount of space, from each other. And dandelions, which are dispersed by the wind, are going to be more random. And elephants are herd animals, so they're going to be clumped. All right, demography, we're back to that. Demography is the statistical study of population changes over time. We're looking at birth rates, death rates, and life expectancies. Let's see, down at the bottom, life tables may include the probability of individuals dying before their next birthday, which is their mortality rate, the probability that they will die before their next birthday. birthday. That's mortality rate. The percentage of surviving individuals dying at a particular age interval. So you may be looking at how many are dying at age 50 versus how many are dying at 80, and their life expectancy at each interval. And then survivorship curves, graphical relationships of populations, number of individuals. Well, this is number of individuals surviving, y-axis, and then the percentage of maximum life expectancy, the percentage that reach maximum life expectancy. So you can see with birds, this is a type 2 survivorship curve. So with birds, the number of individuals surviving, when that is high, the life expectancy is lowest. And when the number of individuals surviving is low, so a smaller population size, you have a higher life expectancy. That's a type 2 curve. So in a type 2 curve, death at any age is equally probable. In a type 1 curve, which humans have, you're going to see death happening primarily in the older years. So this part of the curve is going to be showing individuals are dying, mostly when they're elderly. And then type 3, very few survive the younger years. So that would be like, here it says trees, but like little seedlings, very few actually survive and grow into a tree. Fish are the same way. Turtles, sea turtles are the same way. So these are the 3 survivorship curves. Fecundity is the potential reproductive capacity of an individual within a population. So fecundity, or the potential reproductive capacity, is inversely related to the parental care. What that means is that organisms that give the most parental care tend to have less offspring, and organisms that give more parental care tend to have, I already said that, organisms that that have a large amount of offspring give less parental care. So using plants instead of animals as an example, plants with low fecundity produce few energy-rich seeds. So examples of these plants would be coconuts, chestnuts. Each has a good chance to germinate into a new organism. But if it's a plant with high fecundity, then they're going to have many small, energy-poor seeds that have a relatively poor chance of surviving. So that only a few individuals that are produced actually survive. All right, other fecundity factors are early versus late reproduction. Organisms that reproduce early are likely to produce a larger number of offspring. Sometimes that kills the parent. And organisms that reproduce late, which like humans are an example of that, risk not surviving to reproductive age. So it says reproduce late in life may be able to better provide for themselves and their offspring, but they're taking a risk the longer they wait to reproduce that they're not going to survive to reproductive age. It's not a very high risk with humans. And then we have single versus multiple reproductive events. And I'll come back to this. It's almost 15 minutes.